Hi, everyone. Good morning. So I'm Amlan. I'm a graduate student at the Department of Computational Biology in Cornell University. And today I'll be presenting my work on pre-evasion mechanisms in zebrafishes using interactive experiments. So just to start, why do we even care about predator and prey interactions? So ecologists have been studying these interactions for generation at this point because of how critical they are to population dynamics. But if you look at these interactions on the level of individuals and groups, you see something very, very interesting. So this is data from a recent paper from our lab and collaborators where they collated empirical data of predator success rates in the wild. And what they found was the median of all the data they found was around 0.2 and the mode was almost zero. So you might ask why these predators, which are evolutionarily optimized killing machines, they are often bigger, faster, stronger than the prey they're chasing, not that good at what they're doing. So in short, why are the prey so good at escaping? And this is a question that has piqued the interest of ecologists, mathematical biologists, neuroscientists for some time. So there have been a ton of recent studies on how escapes are There have been a ton of recent studies on how escapes are initiated, and there have been a lot of recent good work on the neural basis of these escapes, like how neurons process these escapes. So, but the typical way that these experiments are usually done is you have a predetermined stimulus that gets played. It could be auditory, visual, or olfactory, and you have some kind of reaction that the prey does, and you look at it and you try to infer what is happening. So typically what you would, if you try to find an analogous version of that in the wild, what you would expect is you might have a predator which approaches a prey and the prey reacts away and the predator actually continues along the trajectory. There is no kind of feedback. There is no interplay between their dynamics. So that is essentially what a lot of experiments have studied escapes in. But I want to ask you that, do you think these escapes ever happen without feedback? So this is video from Curacao from our collaborators of our lab. And oh. So what you're seeing here is, I'm not sure why there is some kind of lag, but so what you see here is a jackfish attacking a chromis fish. So these interactions are not just simple. They have a very complex behavior. You see the fish is reacting to the jack attacking it, and the jack is also reacting to the fish escaping. So actual escapes always have some kind of feedback. There is very rich behavior that happens if you look at escapes in the wild. So what I want to argue today is biomechanics alone does not explain why preys are so good at escaping. Rather, it is the information processing in these feedbacks which determines the escape rates. So typically, imagine you're a fish, you see a big predator approach you. So you decide to escape. There is some neural and biomechanical processing, and eventually there is a motor output. But what I want you to focus on is this. So this motor output actually feeds into the next sensory input. So you are reacting to the predator, and the predator is also reacting to you. So there is a feedback that happens at every single moment. So if you want to study something like this in a lab, you would need an interactive experiments where there is mutual feedback between a prey and also a predator. So we decided to tackle that by building something like this. So we have an arena where we keep our fishes, we test, like all the videos we record are similar to this. So we have these high throughput bachelor cameras, which give a input of like a stream of images to a computer, which runs ultra low latency, state of the art computer vision models, which detects where the fishes are in the arena. And we use these detections and put them into a algorithm which is similar to what predators use in the wild to do something like this. So you have the fish swimming in, and we have a virtual predator that we project using our setup. And as the fish reacts, our stimulus, which is our virtual predator, also reacts to it. So we have this much more complex behavior that is very similar to what I showed you in the video before, like the coral reefs videos. And we record all of this using high resolution, high speed cameras, because a lot of these behaviors actually happen within 20 to 30 milliseconds. So you need to shoot at least 3 to 400 frames per second to get any kind of data. So once you incorporate these feedback into experiments, the prey actually start escaping very differently. So when you do not have a feedback, you have essentially what is known as the open loop system, where you have some stimulus which plays and you have the fish reacting to it, and that's it. But once you have feedback, you have something known as a closed loop system, where the prey reacts, you have the stimulus react to it, and it goes on and on and on. So while you might have a behavior which is similar to this in an open loop system where you have a single escape and there is a baseline velocity that the fish goes after it's done, in closed loop systems, you have behavior which is more like this, where there is not just a single escape, it keeps on happening. There is persistent escapes which happen one after another. So it's not just a single decision, it's multiple decisions all based on each other, which happen one after another. So you have a much richer behavior here. And that is precisely what we are interested in. So we are mostly interested in how do the prey control the decisions to continue fleeing once they have started to flee. So prior studies have told us that what initiates an escape could be a certain number of things, but they never tell you what happens after the initiation. So what happens once you start escaping? How do you decide whether you should continue escaping or whether you should stop escaping? As you saw in the last slide, we get profiles like this, and we are interested in this part of our experiments. Like once the first escape starts, what happens? So we're specifically interested in how do fishes amplify this behavior. We are also interested in how do they terminate these responses. We're also interested in whether the fishes know that they're being chased in some way, and also the computations that underlie this behavior. So we decided to do a couple of experiments where we manipulated certain stuff. So what I'll play a video in a second, but in these experiments, what we did was we chased the fishes, and after a certain moment, like after the fish starts escaping, we actually terminate the stimulus entirely. So you have something which looks like this. So you have the fishes escaping, and the stimulus just goes away after a certain time. And if you look at the velocity profiles of these experiments, you see something like this. You have this is where the stimulus goes away. You have them starting to escape, like there is some kind of higher speeds. So on the y-axis, you have speeds normalized to the body lengths per second of the fish. And on the x, you have the time, of course. And what you see is once the stimulus goes away, the fishes go back down to a baseline velocity. So there is an immediate ramping down of the behavior once the stimulus disappears. But if you think about it, these experiments are not that realistic. Uh, at least in the wild, you never have a case where the predator just disappears entirely after chasing you. So we decided to change this and add something new. So instead of disappearing the stimulus entirely, we decided to decouple this stimulus. So you start chasing the fish, and instead of disappearing, you actually decouple its behavior from the predator, from the prey. That is, like after a certain moment in the experiment, the predator moves as if there is no prey at all. And if you look at the velocity profiles of this across multiple trials and multiple individuals, you see a velocity profile something like this. This looks very similar to the case where you had the disappearing stimulus. So the fishes are still ramping down the behavior. So the virtual predator is still present in the arena with them after the decoupling happens, but the fishes decide to still ramp down the behavior irrespective of this. And if you look at the immediate immediate time period just after these stimuluses are disappear or they decouple, and you try to fit like exponentially decaying function to it. And you try to find these time scales of relaxations, like how quickly do they go back down to baseline velocity. You see that these values, which are in 0.2 and 0.3 seconds, again, very quick, almost the blink of an eye, they're not that different from each other. So the fishes somehow are able to tell that they're not in active danger, even though the virtual predator is there in the arena with them. So that motivated us to do something else. So what could the fishes be using to decide that they should keep on escaping once they've started to escape? So a naive assumption is maybe they're using some kind of threshold. So when the stimulus is below that threshold, you're not escaping. And once the stimulus gets above that threshold, you start to escape. And as long as the stimulus is over that threshold, you keep on escaping. And once the stimulus goes down this plain threshold, you stop escaping. So prior studies in zebrafishes, which is the model organism we study, have shown that the fishes use typically two kind of visual inputs. One of them is called the angular size. And what I mean by that is the relative angle that a virtual predator might occupy in the visual range of the fish. So if the virtual predator is far away from it, far away from the prey, you have a lower angular size. And if the virtual predator is close to the fish, you have a much higher angular size. And the second thing which has been shown in studies is this thing known as looming, which is the rate of change of this angular size that I just described. So when you have an object approach the fish, you will have a positive looming because the rate of change of this angle increase, like since the size of the object increases, you have a positive derivative. And if you have a case where the virtual predator moves away from the fish, you will have a negative looming rate because the size, relative size of the virtual predator decreases as this thing happens. So we decided to look, again, these are sensory, just one second. Again, these are sensory thresholds that were found in papers for initiating an escape in zebrafishes. So once we decided to calculate these thresholds for our experiments, especially the experiments that I showed you earlier of the decoupling. So when the virtual predators gets decoupled from the behavior of the fish. And if you look at these experiments, if there was a threshold, you would expect uh, the threshold, if there was a threshold, you would expect there to be a bit of separation between these two. So what you are seeing here is the perceived angular size input that these fishes are getting. So in these experiments, since we have the coordinates of the fishes, you can find where their eyes are, and you can approximately get the visual inputs that the fishes are getting using simple ray tracing algorithms. So if you look at the visual input the fishes get while they're escaping, and what is the next decision that they do, like do they keep on escaping or do they stop escaping, you get something like this. There is no clear separation in this too, which tells us that maybe they're not using a size threshold at all. So we decided to look for looming thresholds as well. Again, as I described earlier, oh, one second. Oh, there it is. So if you look at looming thresholds as well, you would expect that if they have to keep on escaping, there would be a higher threshold, but we do not find that in our data. There is, again, no clear separation between these thresholds that we calculated. So all of these experiments that we do, they essentially involve two fishes where we target one of the fishes and the other is not targeted. So you typically have velocity profiles like this, where the target fish, which is in the red, starts escaping once the virtual predator arrives in the arena. And the non-target fish, which is in the blue, it does not escape. It is always under a threshold velocity. But if you try to calculate the sensory inputs that I described to you earlier, the angular sizes and the looming, you see something like this. So the non-target fish actually gets a higher sensory input in form of looming and perceived angular size, yet it chooses not to escape, while the fish which escapes has a much lower looming rate and much lower angular size. So what could possibly be happening here? So we decided to look for inspiration in predator behavior. So our lab in this paper that you can see did experiments where we had trouts in a flume tank, and we projected a virtual food particle which moves towards the trout, and the trout has to respond to a last minute change in the perceived direction of this virtual predator. So they have to react very quickly. And we fitted models which the trout could possibly be using based on the visual inputs that it gets to do this kind of behavior. And what we found was the fishes are using a control rule which is similar to this. So what you see here is the rate of turning of the fish, like how quickly can it turn. And we found that it was proportional to the perceived angle of the target in the red, uh, subtracted with the current heading of the fish, which is the h of t. And the way that the functional form which fit the data the best was something like this. Yeah, so what the fishes essentially end up doing for capturing a moving prey is they take the last data that they have, the last position of the target, they extrapolate the target's position using, they calculate these velocities of the fishes, they do a simple linear extrapolation, and they also do this, which is they integrate their own velocity and they try to predict where they will be in the future. So what I mean by this is not only are they keeping track of the relative position of the target, they're also keeping track of their own self motion. They use both these things to at least capture moving prey. So these models where you try to take a current motor output and you try to predict the sensory inputs that you will get in the future are often known as forward models. So they are very prevalent and very popular in motor learning. So what a forward model essentially does is you have some kind of motor input, you have a forward model of that, and you expect some kind of sensory feedback. It's a prediction of your brain of what you should see. And then there is the actual sensory feedback that you get from the environment. And you use the difference between them, this discrepancy between these two things, to update your forward model and to learn. And that's how learning happens. And if I have to put it into a simple model, which a lot of you would actually understand, it would be this. So every time you have gone to a new hotel, like today, and you try to fix the shower knob, what you do is essentially you move it in a way, you expect a certain temperature of water to come, but you get a different temperature. Then you adjust this knob continuously, and finally you find that sweet spot that you really like. So you're using these discrepancies in the sensory feedbacks to also do this unconsciously. So we decided to to check how could fishes be using these forward models for these decisions. So imagine you're a fish. Again, this is probably the third time I'm telling you to imagine you're a fish, but bear with me for a few more moments. So if you're stationary and you see something approach you like this, you would expect a stimulus of looming like this, what you see on the graph below, like you would expect it to increase as time goes. But if the predator is stationary and you decide to move away from it, you would expect something like this. So let me be clear here. What I mean by this here is this is the expectation of what you would see as you move away from a predator. So if the predator is stationary, you move away from it, the angular size changes, you expect a negative looming rate as you move. But could the fishes be doing something like this, where they look at the difference between the sensory feedbacks they get and the sensory feedback they expect? So they could possibly be using something like this, which is the difference between these two graphs. So once this goes above a threshold, maybe they realize that something is chasing them. Again, let me be clear, it's a lot of things on this slide, but the takeaway that I want you to understand here is maybe they're using this difference, this discrepancy between the sensory feedback they expect and the sensory feedback they get. Again, I'm not claiming that this is the exact model they're using. This is just as an example. So all the previous paper that we discussed and the foreign models things that we saw motivated us to do a new kind of experiment. So we wondered whether the fishes can actually tell when they're getting chased specifically. So we decided to do these experiments where we chase one fish, and in the middle of the experiment, we switch the targets. So it is very similar to the trout paper where we switch the direction of the food particle, the virtual food particle. So in these experiments, a typical experiment would go something like this, where you have the first fish, which is target 1 in red, responding to the virtual predator, and the switch actually happens at t equals to 0. So once the switch happens, there is an immediate ramping down of the behavior. And the second target, which is in blue, it starts going ballistic, like they go up to very, very high values, as you can see. So this relaxation and this amplification again happens around in like 0.2 seconds, very, very fast, blink of an eye. So these fishes are somehow very quickly able to say that they're not getting chased, and the new fish is also able to tell that it is getting chased in some way. So we decided to look for data around this switch. So we decided to look for data just before the switch and just after the switch for the fishes, and we decided to calculate these looming prediction errors for them. And if you plot them out, you see something very, very interesting. So earlier, all the plots that I showed you, they do not have separation. But once you account for these prediction errors, you see a much better separation in the data. So again, by loom prediction here, I mean it's the difference between the sensory feedback the fish gets and the sensory feedback it might expect from its own self motion. So you see a much better separation. And again, I'm not claiming that this is the exact functional form they're using. It could be something else. Currently, as we speak, we're taking more data. We're trying to fit more models, and we're trying to figure out what is the exact functional form that the fishes are using. But we believe that we have evidence that the fishes use an error prediction controller instead of it just being a simple threshold. So just to summarize everything so far, the fishes are definitely not using a looming or a size threshold to decide when they should keep on escaping and when they should stop. They are very accurate at determining when they are the targets of a chase. They rapidly shut down their evasive maneuvers when the stimulus disappears or when they're no longer being chased. And they're using some kind of self-motion and sensory feedback to determine when they should keep on escaping. And if everything I told you so far is true, you should be able to mess around with these controllers because you know what they might be using. So we decided to do these experiments where we essentially had no looming. So we had these predators, these virtual predators, which stayed at a fixed distance from the target. So you have no change in the loom because essentially the distance is always constant. So you have a zero loom, you have absolutely zero loom. And when the fish moves, it would expect to see a receding stimulus. So it should expect to see a negative loom, and there is a discrepancy in that. So can you possibly exploit that to make the fishes escape when there is no loom at all? So this is again very recent work. I don't have a ton of videos to show you, but I really wanted to show you this. So we start these experiments with the fish just getting tracked by a virtual predator. But as you can see, once it starts to move, it goes ballistic. Like it might be relaxed at the start, but once it starts to move, it goes absolutely ballistic. This tells us that yes, they're using some kind of error prediction. Like we believe this is the evidence for it. Like once they move all of a sudden, they expect the looming to go down, but when they do not see this looming go down, they start probably realizing that they are in a lot of danger and they start going crazy. So we believe this is early evidence of the fishes using these error prediction models. So just to conclude everything that I have been saying and the broader implications of this work. So all the previous work mostly focuses on the initiation of escapes, but what we are interested in is once this escape starts, what happens, how the brain controls these dynamics of this entire sequence instead of just a single event. We believe ongoing responses seem to demand way more than just raw sensory input. We believe there is information processing, a complex amount of information processing, which determines how they keep on escaping. And again, that is why we believe that these preys are so good at what they do, because they process this information in a non-intuitive way. And most importantly, we believe that the brain may be exploiting these prediction error signals not just for learning, but also for real-time decision making and behavioral control. And that is, I think, the takeaway message for uh, all of you from my talk is the brain simply does not use prediction error to learn something, but it also might be using to do something at the same time. So that's it. And I would like to thank my advisor, Andrew Hine, without whom this would not be possible, fellow members of the Hine Lab, Joe Facho from the Neurobiology and Behavior in Cornell University, and Alex Blazewski from Center of Applied Math in Cornell University. That would be all. Thank you.
Hi, whoa. I was wondering, is there anything in the looming data that suggests when the evasion is successful or not? If there is, like, if they continue evading or stop evading, does
Could you
that
repeat? I
mean?
cannot hear you. Can you?
Oh, yeah, sorry.
Yeah.
Is there anything in the looming data that suggests when the evasion is successful or not?
So the way that we do these experiments is we did a lot of trials and we calibrated these experiments in such a way that there is always a positive loom, but our virtual predator never catches up to the prey, because once it catches up, what would possibly happen is the prey gets under the virtual predator and you cannot see anything anymore and you cannot infer what is happening. So we artificially limit our experiments to cases where the chase is happening, but the predator never actually like intercepts the virtual
Oh,
predator,
I
the prey.
understand. Thank you.
Great talk. So I was wondering, like, is there anything specific to fishes here? Because like what you explained, like I have seen that like a million times in like a National Geographic video, right?
Yeah.
Where you have a leopard chasing a deer and you can see that the deer immediately knows when the chase is off and when it's chasing something else, for example. So is there like scope for like broadening this to other prey predator systems as well, or is this?
There has been, again, the prediction error thing that I told you, that is how a lot of organisms used to learn. And there have been recent papers in zebrafishes which also show that they use it, like they could be using it for something more. And we show in our experiments that they're using it for real-time decision making. So we think that's a take-home message that I was saying earlier. Maybe these organisms are using this general phenomenon of prediction error to do these decisions. So it's not just limited to zebrafish. We believe it could be expanded to other animals, especially the ones which use visual stimulus. Like all the experiments you saw did not involve any kind of auditory stimulus. So anytime you have an organism which uses visual stimulus, we think maybe they're using something similar. So yeah, there is a more general result there. Yeah.
Hi, awesome talk. Thank you.
Thank you.
First thing I noticed in that like introductory video of like the actual wild, all the other fish move to cover at the same time that first pursuit happens,
Yes.
right?
Yeah. I'll just go back to that quickly.
Oh yeah, that might take a.
It's at the start.
There's a lot of good visuals here. But I guess I can continue. These plates, I mean, it's easy to track the fish when there's no when it's just an empty dish. But I was wondering if like incorporating cover is on the horizon, because I bet that changes like, you know, how a fish wants to escape out in the open versus am I trying to make it back home?
Yeah, so what I showed you today was one part of what my lab does, which is experiments in the lab. But there is a second part of the lab, which actually, so the video you saw is what we record in Curacao. So what we do is we have these camera setups where we have two cameras. So once you have two cameras, you can calibrate them and you actually can get 3D trajectories. And the thing is, when you incorporate 3D, it becomes way more difficult. So this is a thing that we actually are doing right now. Like we are trying to see what happens like in the field. Again, it's a very difficult problem. There has been no studies on how to do it. So we're trying to do it at this point. So yeah, we're trying to do that, yes.